Okay, in this lecture, I will be talking about ketone body metabolism. So basically, ketone body metabolism, we'll talk about how it's made and how it's used. So this is different than the other pathways we talked about, mainly because it's synthesized in the liver, but it's usually used by other organs. So overall, it's considered a catabolic process even though part of it is we have to synthesize that, which is usually considered anabolic. So this is usually done in a fasting state. So that's why it's considered a catabolic process. It's made in the liver. Uh, why is this so thick? Let's lower it. Is that better? No. Why is it doing that? Okay, I'm not sure. Okay, so basically it's made in the liver, and it's used by the brain in the muscle. So this is usually used under fasting condition, and it's generated as an energy source. And we'll also talk about that it can also be used as signaling molecules. Okay, so this is kind of just an overview. We have glucose 6-phosphate. We go through glycolysis, and then we make acetyl-CoA. So we already talked about acetyl-CoA. We can go into the TCA cycle. We can make fatty acids, so that would be fatty acid synthesis. We haven't talked about cholesterol yet, but we in order to make cholesterol, we start with acetyl-CoA. And then to make ketone bodies, we also start with acetyl-CoA. So you'll see acetyl-CoA is connected to a lot of pathways. So its regulation is important, and it can connect all these different pathways together and how they're regulated. Okay, so like I said, it's made in the liver and it's sent to other organs. It's just a way for the liver to send energy to other organs. Okay, so this is an overview of it. So the liver can break down tags into free fatty acids, and then those fatty acids, the liver usually doesn't need the energy because liver can run on all different things. It can use glucose, it can use different fatty acids. So usually it needs to send it to other organs who can't rely much on like glycogen, for example. So when we talk about the brain, we'll go over that. So the liver breaks these triacylglycerols into fatty acids, but it needs a way to deliver the acetyl-CoA. So acetyl-CoA, we already said, can't cross the membrane. So we make these things called ketone bodies that are water soluble, that are able to be transported in the blood and then go to other organs. Okay. So, like I said, acetyl-CoA can't cross the membrane. It has to be vented into water-soluble ketone bodies. So, so if we look here, so this is acetyl-CoA in the diagram, and then acetoacetate. Why is it not writing? There. Acetoacetate and beta-hydroxybutyrate are two ketone bodies. We'll talk about three, which can then be converted and then go into like brain or muscle. Okay. So this is a fatty acid being broken down to acetyl-CoA, and then it goes into acetoacetate and beta-hydroxybutyrate. It travels in the blood, and then it goes in here. Beta-hydroxybutyrate can then be converted back into acetoacetate, and then this can be converted back into acetoacetyl-CoA, and then eventually acetyl-CoA, and then this goes into TCA and used as energy. So basically, what I'm trying to explain here is ketone bodies are used to transport energy between organs. Okay, so let's look at the ketone body synthesis. So ketone body synthesis occurs in the mitochondria, and we start with acetyl-CoA. This is important because, so this would be ketone bodies, and then this would be cholesterol. So cholesterol and ketone bodies share one intermediate, which is called HMG-CoA. They don't share an enzyme, but they share an intermediate metabolite. So this is another way that the cell can regulate by location. We already talked about fatty acid synthesis and beta oxidation, how they occur in cytosol and matrix respectively. So this is similar to ketone bodies and cholesterol. Cholesterol occurs in the cytosol, and then ketone bodies occurs in the matrix or the mitochondria. And this is just another way to regulate between these two processes because they share the intermediate HMG-CoA. Okay, so this is just another way to regulate. We'll talk about all these different reactions, how we go from acetyl-CoA to acetoacetate and beta-hydroxybutyrate. Okay. All right, so this is the 3 ketone bodies. So they're all made from acetyl-CoA. And the 3 we make are called acetoacetate, and the 3 we'll talk about. We have acetone, and then beta-hydroxybutyrate. So this can also be abbreviated beta 0HB. So if you ever see that, it's just beta-hydroxybutyrate. You'll notice that this is a ketone. Wait for it to draw. This is a ketone. But then this one is not a ketone, but it's still considered, we have a hydroxyl instead of a ketone here, but it's still considered a ketone body. So they're just called all ketone bodies, even though one of them is not a ketone. Okay, let's look at acetoacetate. So this is the first one synthesized. So this is directly synthesized from acetyl-CoA. This is the main one, and then this one can be converted into the acetone and the beta-hydroxybutyrate. Okay, so the beta-hydroxybutyrate is produced from acetoacetate. We'll talk about why we would convert to beta-hydroxybutyrate instead of just using acetoacetate and transporting that. And like I mentioned, this is not a ketone. Okay, and then we have acetone. So acetone is made of from acetoacetate as well, and it's made via 2 different mechanisms. I'm sure you've all seen acetone before, at least in organic chemistry, at least, or in Gen Chem as well. But it can also be used as a ketone body. Okay, so acetone can be made via 2 different ways from acetoacetate. So I'll talk about that enzymatic way. So it can be made via an enzyme, or it can be made via decarboxylation reaction. So this is a graph. So if we just add acetoacetate in a bunch of serum blood, I guess, some of it will eventually become acetone, because the decarboxylate, let's go back. Acetoacetate just has an extra CO2, so this CO2 over here, more than acetone, but this CO2 can actually be decarboxylated, and it's a spontaneous reaction. So that means it just happens by itself. So if we leave acetoacetate in the blood, it can be spontaneously decarboxylated. So we'll eventually get more acetone over time by itself, because the process is spontaneous. If we add proteins, we'll get more acetone, and this is because the enzyme acetoacetate decarboxylase catalyzes the reaction even more, so it gives us more of acetone from acetoacetate, and this is the reaction over here. It's a decarboxylase. It takes acetoacetate, we decarboxylate, and then we end up with acetone. Okay, so let's talk about what happens under fasting conditions and how that eventually leads to ketone bodies. So under fasting conditions, we'll have low blood glucose levels. So the first thing the liver will do is try to make glucose to export that in the blood. So one way to start gluconeogenesis is to use oxaloacetate. So we use oxaloacetate to convert it into glucose 6-phosphate and then eventually glucose. Then glucose is sent out of the cell to regulate blood glucose levels. So that's the first thing that happens when under fasting conditions. We need to raise blood glucose levels. What this creates is we get low levels of oxaloacetate, low levels of pyruvate. I don't know why I have oxaloacetate twice, but this is supposed to say, yeah, just forget it. We'll get low levels of pyruvate and oxaloacetate because they're used for gluconeogenesis. What ends up happening is because all of the oxaloacetate is being used under fasting conditions, the liver will also start breaking fatty acids, so we'll get more acetyl-CoA. Because we're getting a lot more acetyl-CoA and oxaloacetate is being used to gluconeogenesis, this acetyl-CoA will start to accumulate. And what this does is this signals the body to export the acetyl-CoA as ketone bodies. And that's where we get ketone bodies from. We have a buildup of acetyl-CoA, and liver needs to do something with that, so it makes into ketone bodies and sends it to other uh, organs that need the energy. So just to summarize, gluconeogenesis is used to regulate glucose, blood glucose levels. Acetyl-CoA is converted to ketone bodies as a way to export the energy. Because we're preserving oxaloacetate and pyruvate for gluconeogenesis, this causes a buildup of acetyl-CoA because the TCA cycle is not running as fast. So yeah, since oxaloacetate is being used for gluconeogenesis in low concentration, it causes buildup of acetyl-CoA, and then that's why we make ketone bodies. Okay, so hopefully this connects everything together that we talked about. Okay, so let's talk about how it's exported. So we have, this is a fatty acid, it's acyl-CoA, we enter in the mitochondria, so this is the mitochondria. All right, this is really bothering me, how like thick this is. No, they're all the same. Okay, I think this is better. Or whatever. I don't want to waste too much time trying to fix this. Okay, so let's talk about how ketone body, acetyl-CoA is eventually exported as ketone bodies. So we have acyl-CoA here, comes inside the cell. We convert it into acylcarnitine, we enter the mitochondria, and then we eventually make acetyl-CoA. So we need to send this energy to other organs. So if we run the citrate shuttle, which we talk about when we went over fatty acids, we convert to citrate, then we convert citrate in the cytosol, and then we convert back into acetyl-CoA. But, so this is the cell membrane. So we already talked, all right, this is a lot worse. So this is the cell membrane. And we already talked about that acetyl-CoA can't cross the cell membrane. So we need a way to make, to transport the acetyl-CoA as energy for other organs. So what ends up happening is we don't even run the citrate shuttle. We end up converting acetyl-CoA into ketone bodies inside the mitochondria, and then they can cross both membranes, enter in the blood, and then be sent to other tissues. So yeah, like I said, acetyl-CoA is converted to water, soluble ketone bodies, and then be exported in the blood. So I just added this as a review. Wait for it to load because we talked about it in fatty acid synthesis. It's been a while, but this is the citrate shuttle that I talked about. So this doesn't happen. I'm just gonna write down doesn't happen because we can convert the acetyl-CoA into because a lot of time you think you run this reaction and then you make acetyl-CoA in the cytosol and then convert into ketone bodies. So this doesn't happen. Acetyl-CoA can be doesn't need to go into the cytosol. It is converted into ketone bodies straight in the matrix, and then from the matrix, it is sent in the blood. So this is the citrate shuttle. I'm just going to go over it briefly. We have pyruvate. It's made into oxaloacetate. So this is pyruvate carboxylase. Talked about it in gluconeogenesis, and then oxaloacetate is converted to citrate. So this is done by citrate synthase, which is the first enzyme of TCA, and then we use the tricarboxylate system. So this is where you get the citrate shuttle from or whatever. And then citrate is sent in the cytosol, and then we eventually get acetyl-CoA back in the cytosol, and then we convert it into oxaloacetate, and then using malate dehydrogenase. So this enzyme is in TCA, it just runs backwards in the cytosol. We make malate, and then using malate enzyme, we convert back to pyruvate, and then pyruvate is sent into the mitochondria. So this does not happen. This is just, it's a common misconception. That's why I added it. The acetyl-CoA uses this to get into the cytosol and then made into ketone bodies. So ketone bodies happens in the matrix, so we don't need to run this. Okay? So this is acetyl-CoA. Our transport mechanism is going to be converting it into acetoacetate, so this ketone body, into the matrix, which then can be exported. Next slide. Okay. So ketone bodies is used under fasting conditions, and like I said, it's mainly used by the muscle and the brain as an energy source. I think I don't know if you'll be asked about this, but sometimes the heart can use it and other peripheral organs. But if asked, the heart can do some ketone bodies. But we're mainly gonna talk about the brain, but the muscle also uses it as well. Okay, let's talk about why the brain would use. We're only gonna talk about the brain, not the muscle. So let's talk about why the brain would use ketone bodies over glycogen or breaking down of fatty acids. So this is the blood-brain barrier, and what it does, it makes it really hard for stuff to cross in. So the blood-brain barrier prevents molecules from crossing the brain. This is just made to protect the brain from and keep things out of the blood from entering the brain. So this is just a mechanism that the brain developed. So there is we know that plasma components from the blood can diffuse into the brain, and then these molecules. So these are plasma components in the blood. So these molecules end up in the astrocytes. Uh, so if we look at the blood and the components of it, we'll see the plasma components enter the endothelial cells, and then they eventually they go through the astrocyte first before they enter the neuron. So you can see that it can be really hard for stuff from in the blood to end up in the neurons. So fatty acids, for example. So let's talk about why the brain cannot use fatty acids. Fatty acids are, if they enter through the blood-brain barrier and cross, they'll end up in the astrocyte, and then the astrocyte will end up using them. So this is one reason that the neurons of the brain don't can't really run on fatty acids, and we'll talk about the two other ones because it's limited by the vascularity of the brain, and then less of it can enter into the astrocyte in the brain because of the blood-brain barrier. So just to review, this is the neuron, the astrocyte. Basically, they function as one unit. I just added this as a review. The astrocyte stores the glucose as glycogen, and the neuron does all the action potential and all of that stuff in the brain because that's its main job. And they can exchange stuff through lactate, glutamate, and glutamine. And then glucose can enter from the blood into both. So the only thing that they can really enter through this blood-brain barrier that's easily enter is glucose. But then when under fasting condition, we have low blood glucose levels, so we can't really use that. And then fatty acids would take too much time to cross the blood-brain barrier and then end up here. So we need a different way. Okay, so let's talk about. So this is just another way to compare them. So this is the blood vessel. You see it's carrying glucose. So this is oligodendrocytes. So those are the ones that make myelin for neurons. This is the astrocytes, which mainly stores the glycogen that's used under when the neuron needs energy, and this is the neuron. Okay, so there's two different stages of fasting. There's like the initial one where I guess I forgot the like official name for it, but it's just like when you're hungry, and then the second one is when you're like fasting. That's when you haven't eaten in like a really long time. So when the neuron starts running out of energy, eventually the astrocytes will start breaking down its glycogen into glucose, and then you'll make pyruvate, you'll make lactate, and then you send the lactate into the neuron. Okay. Eventually, the astrocyte will run out of glycogen, and then this is the stage we're talking about when we say fasting stage. So there's starvation, and then there's fasting. Fasting stage is after the astrocyte broke down all of the glycogen. So this, we're already out of glycogen. We'll talk about why we can use fatty acids. So we need a different way to supply energy for neurons. Okay, so like I said, glycogen is stored in the astrocyte, but it's stored under. It gets very low when you get into like the fasting condition, and then eventually the starving it gets very low in during fasting conditions because it's used up in the starving conditions. And we also can't store as much as glycogen as liver. So in the astrocyte, we can store 3 to 12 We can store 3 to 12 moles of glycogen per gram of tissue. And in the liver, we can store 100 to 500. So you can see the astrocyte stores way less glycogen than the liver. So eventually, after a little bit, the astrocyte will run out of glycogen, and then we need a different energy source. Okay, so can the brain do fatty acid oxidation? It could, but it usually doesn't for multiple reasons. The reason is it's called fatty acid oxidation, so we're doing a lot of redox, oxidation reduction reactions. What this does is increases the risk of reactive oxygen species, and those are really bad. If you when you look at apoptosis and how that happens, it usually results from a lot of reactive oxygen species. So reactive oxygen species can kill the cell. And neurons don't divide; they're already mature. And so if you kill a nerve cell, you can't regenerate it because your nerve cells don't divide. They're not like your liver cells. So it doesn't want to run fatty acid oxidation because that increases the risk of having to deal with reactive oxygen species and then eventually killing neurons or nerve cells. And another reason is it's too slow. We already talked about having to cross the blood-brain barrier. After that, it has to cross into the cell membrane. Then you have to go into the mitochondria. And then you have to go through oxidation. So it's kinetically a lot slower than using pyruvate, for example. So we need some sort of way to give the neurons energy. That's where ketone bodies come into play. Once the liver makes them, we send them in the blood, and then ketone bodies can be converted right into acetyl-CoA, which can then enter TCA really quickly. So this is kind of why the brain uses ketone bodies. Muscle also uses it for some of the reasons. We only talked about the brain in here, and I think you only need to know the brain. Ketone bodies just serve as a carbohydrate source of energy for the brain under fasting condition. That's basically what I'm trying to say with all of this. Why we use ketone bodies over breaking down of glycogen or breaking down of fatty acids. Okay, because it can enter right into TCA, and then TCA can run and then give us energy. I will mention, we'll talk about it later. It can also it has a lot of effects on gene transcription, and some of them are really positive, and there's a lot of research going on with that right now. Okay, so let's look at the reactions of what happens. So this is the synthesis of ketone bodies. So this happens in the liver. Okay, so although we are synthesizing ketone bodies, the whole process is considered a catabolic reaction, and it is run when under fasting conditions, when energy in the body is low. So don't confuse that, because just because you're synthesizing something does not mean you're in a high energy state. The liver synthesizes this because the liver has energy to synthesize it, and then it's able to send it to other organs who need it more, which then are able to break it down. So ketone body metabolism is considered a catabolic, not catabolic. It's a pathway that is run under low energy. So whenever you're talking about all the the stuff that's run under low energy, like fatty acid breakdown or glycogen breakdown, ketone bodies will be in the same boat. Okay, so let's briefly talk about it. So we have acetyl-CoA. We get 2 acetyl-CoA molecules, and then, like I said, it's synthesized in the mitochondria. We get 2 acetyl-CoA molecules, and then we make acetyl-acetyl-CoA. So this is done by thiolase. Thiolase, so this is if you remember from beta oxidation, this is the reverse of the last step of beta oxidation. It's the same enzyme, same everything. And it happens in the matrix as well. And then after that, we have HMG-CoA synthase. So this takes a third. So this is a third acetyl-CoA. So we take a third acetyl-CoA, and then HMG-CoA synthase makes HMG-CoA from it. And then HMG-CoA lyase eventually breaks that molecule into it causes a cleavage, and then it makes it. We break it into two different molecules. We make acetoacetate, and then we make acetyl-CoA. So this acetyl-CoA can then go back and be used as number 3, the third acetyl-CoA in the next ketone body that is being made. So technically, we only need two acetyl-CoAs to make one acetoacetate, because the third one that we use, that that comes from the product of a previous set of reactions that run before it. So don't confuse that, because we make 3 acetyl-CoAs. We use 3 acetyl-CoAs, but because we make one, technically we only need 2 acetyl-CoAs to make acetoacetate. Okay, so let's look at the first reaction. So this is the same as the last enzyme in beta oxidation, but in reverse. It takes 2 acetyl-CoAs. We join them together, and then we lose this thioester bond. Not thioester, because it doesn't have a carbon. This sulfur bond with that has the CoA on it. And then we produce a beta keto thioester, which is called acetoacetyl-CoA. So this is a thioester. After that, we run through HMG-CoA synthase. So it takes acetoacetyl-CoA from the previous reaction, and then we make HMG-CoA. So this is the rate limiting step. It's done by HMG-CoA synthase. All the other reactions are reversible. It's also important that because it's also important because HMG-CoA is shared with cholesterol. It's a shared metabolite with cholesterol metabolism. So, like I said, it's important to regulate between that and cholesterol. So that's why ketone body synthesis occurs in the matrix of the mitochondria, and then synthesis of cholesterol occurs in the cytosol. So that's one way to regulate between those two. Like I said, we take HMG-CoA, and then HMG-CoA lyase comes in, and then cleaves it into acetoacetate and then acetyl-CoA. Okay, so they're pretty straightforward, just 3 reactions. And then this acetyl-CoA, like I said, can then go back and be used in the second step of a different acetoacetate that is being made. Okay, once we make acetoacetate, we can convert it into the 2 other ketone bodies, which is acetone and beta hydroxybutyrate. So we already talked about the acetoacetate to acetone, so this can happen spontaneously, or we can run the acetoacetate decarboxylase reaction. And then acetoacetate to beta hydroxybutyrate, this is done by beta hydroxybutyrate dehydrogenase. Okay, this is this reaction also. So this is I'm going to talk about why we convert acetoacetate to beta hydroxybutyrate, because why can't we just run acetoacetate and then send that in the blood and then convert it right back into acetyl-CoA? We'll have less reactions. The reason we convert into beta hydroxybutyrate and then convert back when we get to the target tissue is because we use NADH. We use NADH to convert from acetoacetate to beta hydroxybutyrate, and then we make NAD+. So this is in the liver. So the liver uses NADH. This is important because when we get to the target tissue, so this would be target tissue. So this would be your muscle or the brain. We can run the reaction backwards. When we run the reaction backwards from beta hydroxybutyrate back into acetoacetate, right? We regenerate NADH. Okay. Why do we need to regenerate NADH? Because NADH is low. We're in fasting conditions, right? So we're in the low energy conditions. So you'll have a lot more NAD plus than NADH. So we need a way to regenerate electron carriers. So this is the reason why we can't just send acetoacetate directly into target tissues. We can convert it into this intermediate using NADH, and then we make NAD in the liver, and then when it gets to the target tissue, we regenerate NADH back for the target tissue. So this is just a way to transfer electron carriers between organs. So if you ever get a question about that, so this would be the perfect example. convert it into beta hydroxybutyrate, and then once it gets to the target tissue, we convert it into acetoacetate. This is similar to how the Cori cycle between the muscle and the liver, where we send lactate, eventually made us, we send lactate from the muscle, then it's converted into pyruvate, and then we run gluconeogenesis and the cycle continues. So this is very similar to that. This is important because if you ever asked why we can't just send acetoacetate and why do we need to use beta hydroxybutyrate, this is one way why we use the beta hydroxybutyrate. Okay, so this is once acetoacetate gets into the target tissue, or if beta hydroxybutyrate gets to target tissue, it's converted to acetoacetate, and then once it does that, it runs through a few more reactions to eventually get acetyl-CoA. The first reaction we use, we take acetoacetate, we take succinyl-CoA, and then we make acetoacetyl-CoA, and then we also regenerate succinate. This reaction is ran by an enzyme called, you can abbreviate it as SCOT, and it's short for 3-ketoacyl-CoA transferase. It's important, this reaction is coupled with succinyl-CoA going into succinate, so this is your DCA stuff, except we don't generate GTP. Because you're not running it coupled with the complex 2, you're just using it as a carbon donor. Not as a carbon donor, we're just using the succinyl-CoA to attach this S-CoA into acetoacetyl. So because we're not coupling this to complex 2, we don't generate GTP in this process. So it's important to look at that, because just because succinyl-CoA is going to succinate does not mean we're automatically generating GTP. And then once acetoacetyl-CoA is made, we run into thiolysis, and then we regenerate acetyl-CoA back. So the rate limiting step is the SCOT enzyme, and it's important because this enzyme is expressed only in expressed in the periphery tissues that use ketone bodies, the brain and the muscle, and it's not expressed in the liver. The reason is because the liver is what makes ketone bodies. If we express this enzyme in the liver, we'll just have a futile cycle. We'll be making ketone bodies in the liver and then breaking them down in the liver. So it's important that the liver only expresses the enzymes that make the ketone bodies, and the muscle and brain express the enzymes that can break down the ketone bodies. And it's mainly done by this rate-limiting enzyme SCA. And like I said, thiolysis takes acetoacetyl-CoA, and then we make 2 acetyl-CoA molecules. So we regenerate the 2 acetyl-CoA molecules that we used in the liver to make our ketone bodies. So we successfully transferred energy from the liver into the target tissue. Okay, so let's talk about some regulation of ketone bodies. Okay, so the first one is the liver doesn't express SCOT. So the liver makes the ketone bodies. It doesn't want to express the enzyme that breaks them down, so to avoid futile cycles. So that's one way it's regulated. Second way of regulated is the level of free fatty acids in the liver. And this can also come from adipose tissue. Because ketone body synthesis starts with acetyl-CoA, and fatty acid breakdown gives us acetyl-CoA, the level of free fatty acids available will dictate how much ketone bodies we can run. And this usually comes from ketone bodies or any fats that are stored in the liver. Another way to regulate ketone bodies is through the glycerol 3-phosphate shuttle. The glycerol 3-phosphate shuttle uses glycerol 3-phosphate, which is used to make triacylglycerols. So if we have a lot of glycerol 3-phosphate shuttle activity, we have a lot of glycerol 3-phosphate, which means we have a lot of triacylglycerols, which can then be broken down into individual fatty acids and then eventually ketone bodies. So high levels of that will allow us to synthesize more ketone bodies. Also, for the TCA can regulate ketone body use, because we use acetyl-CoA in TCA, and then we also use ketone body. We use acetyl-CoA in making ketone bodies. So if TCA is using acetyl-CoA as an energy source, this will inhibit ketone body synthesis because we don't have our starting point for ketone body synthesis. And then if oxaloacetate is being used, so I already talked about this part one. Oxaloacetate is being used in gluconeogenesis. Fatty acid oxidation will build up and then eventually break down into acetyl-CoA. Oxaloacetate is in low concentration because it's used in gluconeogenesis. Acetyl-CoA concentrations will increase, and then acetyl-CoA needs to go somewhere, so it will eventually be converted into ketone bodies. So TCA activity and gluconeogenesis activity will regulate how much ketone bodies we make in the liver. So those are different ways that can regulate how much or how little ketone bodies can be made. Okay, so let's look at regulation. So I will say this can get a little complicated when I talk about it. So mTOR, let's start with mTOR first. mTOR signals nutrient availability. So this will be active after like you eat a meal, for example. You're you're full, you have high blood glucose levels. So what it will do is it wants to inhibit ketone genesis because we said ketone body metabolism occurs under fasting conditions, under low energy states. So this is another way that it can be regulated. And the way that it does that is actually I have a specific slide for it. Basically, we already said the HMG CoA synthase is the rate limiting step. So it regulates through all these different transcription factors to eventually inhibit ketogenesis. So like I said, it's a kinase, and it increases in like anabolism, like for example would be glycogen synthesis or fatty acid synthesis. So it signals nutrient availability. Well, I'll talk about these in just a sec, because I want to talk more about mTOR. So mTOR can form two different complexes. Can form mTOR-C1, which is mTOR with a bunch of other proteins attached to it. I don't think you need to know all of these. And it can also form mTOR-C2, which is a bunch of other things attached to it. They both signal nutrient availability. So basically, when they're active, they'll activate genes that increase anabolism and decrease catabolism. So when mTOR C has low concentration levels, that means this will increase ketogenesis, because that means they're not signaling nutrient availability. That means we're in a starving state, so we'll increase ketogenesis. So let's briefly look at mTOR C1. So some of the pathways that does. So remember, it signals nutrient availability, so the cell has a lot of energy. So it will eventually activate stuff that upregulates translation. We'll translate a lot of proteins. We're in a high energy state. We'll make more ribosome biogenesis, and then I don't know what this is, but it will upregulate that as well. Okay, so let's look at mTOR C2. So what this does is it also signals nutrient availability. So what the cell will do is it will do proliferation. It will just start replicating when it has high energy state. It also upregulates genes for survival and metabolism. Okay, so let's go back to this slide and how mTOR C1 is related to HMG CoA. Okay, so there's this transcription factor called PPAR alpha, and what this does is it associates with HMG CoA synthase. So HMG CoA synthase and PPAR alpha associate together, and they increase the transcription of each other. Okay, so HMG CoA synthase acts as an activator of PPAR alpha, and PPAR alpha can increase the transcription of each. When I say that is, both of these are proteins, but they can increase the transcription of each other's DNA. Okay, so it's important to keep DNA and protein separated when we're talking about this. Okay, when you add succinylation or acetylation between them. This will reduce their association and decrease their association together, so they won't be able to activate each other. And palmitoylation increases the association between them, so then they'll be able to activate each other. Okay, so mTOR, because this signals nutrient availability, how it acts on ketogenesis is it inhibits PPAR alpha. When you inhibit PPAR alpha, you're not transcribing it. It's unable to associate with HMG-CoA syntase, so you'll have less HMG-CoA syntase because you're inhibiting an activator of HMG-CoA syntase. PPAR alpha acts through different things. It acts through on FGF21, and then that eventually increases beta hydroxybutyrate production. Okay, so that's how mTOR C1 can inhibit ketogenesis. It acts on PPAR alpha. Beta hydroxybutyrate, which is a ketone body, a product of ketone body metabolism, so this can actually inhibit a histone deacetylase. Okay, so a histone deacetylase removes acetyl groups. So this is the histone deacetylase of the gene for PPAR alpha. The histone deacetylase, what it does is it removes acetyl groups from the histones. What this does is it allows the histones to bind the DNA more tightly, so you're transcribing less of it. What eventually it does is the HDAC eventually inhibits PPAR alpha because we're removing these acetyl groups. Beta hydroxybutyrate, a ketone body, like I said, HMG-CoA syntase and PPAR alpha are activators of each other, so they're activating each other. And beta hydroxybutyrate is a product of a subsequent reaction after HMG-CoA syntase. So what it does is it inhibits beta hydroxybutyrate inhibits this HDAC to lead in. So eventually we have more PPAR alpha. So we're inhibiting an inhibitor using beta hydroxybutyrate. I'm going to draw it in red. So we're inhibit this inhibitor because HDAC inhibits that. So we eventually lead to more PPAR alpha that is being made. And then when more PPAR alpha is being made, it's able to associate with HMG-CoA syntase. And then all of this would eventually... And then PPAR alpha can also act on other things because it's just transcription factor. And then all of this will eventually lead into more beta hydroxybutyrate production. This can be really hard. I remember it took me a while to understand everything that's going on, and probably won't understand it the first time. Might have to watch it a couple times or read the book about it. But I tried my best to explain it. Okay, I'm just gonna briefly talk about. I already mentioned this, but I'll briefly review this from genetics: histone acetylase transferase, and then histone deacetylase transferase. So they're abbreviated as HATs and then HDAC. HATs, what they do is they add acetyl groups, and then what acetyl groups are neutral, and if we remember, DNA is negatively charged. Histones tend to have a lot of lysine, which is an amino acid that has a high positive charge. So histones and DNA associate with each other because they have this positive and then the negative charge, and they create this really tight bond, and then it allows DNA to be in a closed conformation so it's not transcribed. So when the HATs come in and add the acetyl group, they make the histones more neutral, so they associate less with the DNA, and then we eventually increase transcription. HDACs then do the opposite. They remove the acetyl group, and then when we remove the acetyl group, the bond between histones and DNA is stronger, so then this inhibits transcription. So when histones are bound to DNA, this is a closed conformation, and then we repress transcription. When histones are not bound to DNA, we have an open conformation, increased transcription. And then these two act on these things to be able to change it. Let me erase all of this. Okay. So that's just a brief review of histone acetylase and histone deacetylase if you guys forgot it. Okay, so I already mentioned that ketone bodies can inhibit HDACs of the PPAR alpha. They can also inhibit histone deacetylase of other things. So this is a beta hydroxybutyrate. So it can go into the mitochondria. So this would be ketone body usage. By the way, ketone body metabolism, where it's made and where it's synthesized are both in the matrix, are both in the mitochondria. So it's synthesized in the mitochondria of the liver, and it's used in the mitochondria of neurons or muscle cells. So I don't think I've mentioned that it's used in the mitochondria, but it's used and made in the mitochondria. It's converted into acetyl-CoA, which is eventually converted to citrate, and then citrate can then be used in other things. So it can act as a second messenger system and bind GPCR proteins on the surface of the cell. So it can activate different pathways and inhibit different pathways. Eventually, it will lead into more cyclic AMP and more calcium. It will activate pathways that lead into more of this and inhibit ones that decrease cyclic AMP. And then eventually we get lower metabolic rate, reduced lipolysis, so we're reduced fat breakdown. And then we reduced sympathetic tone. And then it can also act on histone deacetylase. So remember, histone deacetylase inhibit transcription because they remove the acetyl group. So it inhibits an inhibitor. And what this does is it increases transcription of really important genes. Some of these genes include ones that used for resistance against oxidative stress. So that would be ones against reactive oxygen species, which is good for the cell. It can also, so this is the different pathways. It can go into the cell, be converted into acetyl-CoA, and then citrate. Citrate is then exported, and then this acetyl group, right? So what do histone acetylase transferase do? The hats, so this is hats. They increase transcription by adding acetyl groups to lysine. So we can use this acetyl group from acetyl-CoA that's made from beta hydroxybutyrate to be attached directly on the lysine. So this is both, so it can act on HDAC in this way, and it can be used as the acetyl donor group for histone acetyltransferase to be used. So that's how beta hydroxybutyrate can act to increase transcription of genes. Okay. There's also a lot of research going into this that ketone bodies allow us to make glucose from fats. Because we break the fats in the liver, we make acetyl-CoA, and then eventually one of the ketone bodies that we convert to is acetone. And then there's a lot of research that acetone can be converted to lactate through this methyl glyoxyl intermediate. And then this uses a cytochrome P450 enzymes. There's a lot of research into this because this can help with a lot of the obesity. In the United States, if we're able to make glucose from fats, so there's a lot of research on like upregulation of what enzymes or whatever to help with this process and stuff. Just added it because I thought it was cool. Okay, so let's talk about a condition that can happen when you make a lot of ketone bodies. So this is called diabetic ketoacidosis. So this occurs in type 1 diabetes. So type 1 diabetes, they have it's an autoimmune disease. Basically what happens is you destroy the alpha cells or beta cells. I forgot the ones that make insulin. I forgot which ones they are. I think it's definitely alpha cells. Let me look it up. Actually, alpha cells make glucagon. So, beta cells make insulin. Sorry about that. So, it's an autoimmune disease where the beta cells of the pancreas. Why can't I remember any of this? Yeah, it's in the pancreas, I think. Right. Yeah. Right. Beta cells of the pancreas. Why can't I remember this? Uh, where the beta cells of the pancreas are destroyed, it's an autoimmune disease, so we can't excrete insulin. So insulin is unable to have its effects, and then this eventually will lead to a lot of glucagon. This was say glucagon. And then what this glucagon does is it signals. Remember, insulin signals high energy state, and glucagon signals low energy state. So all this glucagon that is being excreted will signal low energy state, even if we're in a high energy state. One of these things that will be made is ketone bodies, because ketone bodies are made during low energy state. Some of the symptoms are hyperketonemia, metabolic acidosis, and hyperglycemia. So hyperglycemia is high blood glucose levels because we don't excrete insulin, so we're not telling the cells that we're in a high energy state. We keep telling them we're in a low energy state because we have more glucagon, and glucagon signals low energy state, so then the liver runs gluconeogenesis and keeps sending glucose into the blood, and then we eventually lead into hyperglycemia. And then because insulin is unable to bind and tell the cells to use the glucose is just in the blood by itself. So this is diagnosed by something called an anion gap ion selective electrode, which measures cations and anions in the blood, and they should be equal. But in this condition, the cations are more. And what happens is when we excrete ketone bodies, large amounts of Na and potassium plus are excreted in the process. And how does this lead to ketoacidosis? Let's talk about the buffer real quick. When the Na and the potassium are excreted, when ketone bodies are excreted, Na plus and K plus or potassium are excreted as well. What they do is they react with bicarbonate, and they lower its concentration. So this is bicarbonate plus H plus. They bind with bicarbonate and react and form different compounds. What this does is this lowers the concentration of bicarbonate, and our bicarbonate system shifts to the left because we have lower bicarbonate, and then we'll eventually make more H plus. If we make more H plus, this will lower the blood pH levels, and this is why we get acidosis. So I think there's one question on the homework that talks about how is this condition leads to acidic blood levels. Basically, because under this condition you don't make enough insulin, so and you make excess glucagon. This glucagon tells your body to make ketone bodies, and then the ketone bodies are excreted. Some of the Na plus and K plus are excreted as well. They bind through bicarbonate and then eventually lead to more H plus, and then this lowers the pH of the blood because you have more H plus from the bicarbonate system. Okay, so we talked about that ketone bodies can act. We have a couple more slides, I think. Yeah, just almost done. How long is it? It's 53 minutes. Okay. So we already said ketone bodies can act directly on or indirectly. So indirectly, we already said they inhibit HDAC because HDAC can inhibit a lot of the genes, like reactive oxygen species genes. So they can act indirectly on that, or they can act directly by serving as the acyl group for HAT, the histone acetylase transferase, to use to increase transcription. So the acetyl or acetyl-CoA or any of those, they have the acyl group. So what they do is they take the acyl group from that group from the ketone bodies and attach it to lysine, and then we get acetylysine. So we have a neutral histone that's unable to bind the DNA. So then this increases transcription. So that's how they directly increase transcription. And then the indirect way is by inhibiting HDAC. I will mention because succinyl-CoA is coupled with succinase, so this is the enzyme SCOT. We can add a succinyl group to lysine as well. So we always talk about adding a acetyl group. We can add a succinyl group to lysine, and this will have a similar effect as adding acetyl group to lysine. They both make lysine more neutral and eventually increase transcription of genes. And this lysine residue is on histones, by the way. I just assumed that. But when we're talking about histones and stuff, lysine residue is on the histones. So indirectly, would be inhibiting HDAC. So we already talked about this. They can inhibit class 1 HDAC. There's a bunch of HDACs, and they each deacetylase different genes. Class 1 HDACs, basically they add the acetyl group on the lysine, like I mentioned. The association between the histones and DNA is then lowered, and then DNA is able to be transcribed. Class 1 HDACs are involved with P53 and c-MEC. If you're in cell phys, you've probably seen those, but basically P53 is a tumor suppressor gene, which is a good gene. This is one of the reasons why people talk a lot about like ketone. What is it? The keto diet, and why ketone diet is good. Because it basically increases transcription of a lot of the good genes, like the P53. And some people believe that it can regulate lifespan. And like a lot of this is very speculation, but there's a lot of research on that. But because it increases transcription of protective genes involved in reactive oxygen species, and P53, which is a tumor suppressor genes, that's why there's a lot of that's why you've seen like the surge in keto diet this last decade or so, because they increase transcription of really good genes. Okay, so in summary, ketone bodies, so you have acetone, acetoacetate, and beta hydroxybutyrate. Those are the three. They're made in the liver, mitochondria of the liver. They provide fuel for the brain muscle. They're converted into acetyl-CoA into the matrix of these peripheral tissues, and then they are used as energy. And then they can enter directly into TCA for energy. Overproduction can lead into acidosis because you shift the bicarbonate system to the left. When you make all these cations, when you excrete a lot of ketone bodies, they can also function as signaling molecules, like I said. So they can bind GPCR or HDAC. So they can act as signaling molecules and affect transcription through all these ways. So that's it for this video. I said I'm gonna do cholesterol metabolism, but I think I will do energy regulation first. I'll try to get it done this week. I know I'm really behind, but it's fine. I had a lot of tests and stuff going on, but I'm gonna try to get the next ones done in the next few days. All right, thanks for watching.